This is Ravi Jindal and Sarah McWhinney's Explain Everything for Cellular Respiration. The overall equation for cell respiration is 1 glucose plus 6 oxygen molecules creates 6 CO2 molecules, 6 waters, and 36 ATP. The whole reason for cellular respiration is to create ATP from ADP and inorganic phosphate. ATP is the primary source of energy for almost all living organisms. The first stage of cell respiration is glycolysis. Glycolysis means sugar splitting and occurs in the cytoplasm of the cell. The only reactant for glycolysis is glucose, and all of the reactions are catalyzed by specific enzymes. The products of glycolysis are 2-pyruvate, 2-NADH, and 2-ATP. The first step of glycolysis is the energy investment stage. 2 ATP are required to get the process of glycolysis started. One of the 2 ATP attaches to a phosphate group on glucose to create glucose 6-phosphate, or G6P. G6P then rearranges itself into fructose 6-phosphate, or F6P. The second ATP attaches to a phosphate group on the end of the F6P molecule to create fructose 1,6-bisphosphate, or F16BP. What this equates to is each of the 2 ATP molecules giving a phosphate group to the 6-carbon backbone. The 6-carbon backbone of fructose 1,6-bisphosphate then breaks down into 2 3-carbon backbone molecules. One called glyceraldehyde 3-phosphate, or G3P, and one called dihydroxyacetone phosphate, or DHAP. Each of the 2 G3P molecules created in the last step then give a hydrogen and 2 electrons to NAD+, which then reduces into NADH. The 2 G3P molecules each combine with an organic phosphate to create an unstable molecule called 1,3-bisphosphoglycerate, or BPG. Because of its instability, 1,3-BPG quickly gives up its recently gained phosphate group to ADP. This creates ATP through a process called substrate-level phosphorylation. This leaves the 2 molecules of 1,3-BPG with only one phosphate group. This gives them the new name of 3-phosphoglycerate, or 3PG. 3PG then quickly rearranges itself into its structural isomer 2-phosphoglycerate, or 2PG. Both of the 2PG molecules then give up one hydrogen ion and one hydroxyl group, which combine to form water. The resulting 2 molecules are called phosphophenoPyruvate, or PEP. Like 1,3-BPG, PEP is a very unstable molecule and gives up its phosphate easily. This is the second occurrence of substrate-level phosphorylation. PEP readily gives up its phosphate group to ADP, creating ATP. This marks the end of glycolysis. The second stage of cellular respiration is called pyruvate oxidation. This occurs in the matrix of the mitochondria and requires the 2-pyruvate created from glycolysis. This set of reactions produce 2 acetyl-CoA molecules and 2 NADH coenzymes. To enter the matrix of the mitochondria where pyruvate oxidation occurs, a membrane carrier is sent to bring the pyruvate molecules through the outer and inner membranes of the mitochondria. At the beginning of pyruvate oxidation, coenzyme A connects to the acetyl group of the pyruvate molecule seen here. What this allows for is a redox reaction to occur, where pyruvate is oxidized and NAD+ is reduced into NADH. The remaining carboxyl group is removed as CO2 waste, seen here. The product of this redox reaction is acetyl-CoA. This marks the end of pyruvate oxidation and the beginning of the Krebs cycle. The third stage of cellular respiration is Krebs cycle. Like pyruvate oxidation, it occurs in the matrix of the mitochondria. The two acetyl-CoA molecules created in pyruvate oxidation are the reactants for this set of reactions to create oxaloacetate, 2 FADH coenzymes, 6 NADH coenzymes, and 2 ATP molecules. The first step in the Krebs cycle is a reaction between acetyl-CoA, here, and oxaloacetate, here. This creates citrate, seen here, with the coenzyme A being removed. Citrate undergoes an isomerization to form isocitrate, which is required for the next step of the Krebs cycle. Isocitrate, seen on the left here, undergoes a redox reaction where it is oxidized into alpha-ketoglutarate. An NAD+ ion from the mitochondrial matrix is reduced into the coenzyme NADH. In the fourth step of the Krebs cycle, alpha-ketoglutarate, seen on the left, forms succinyl-CoA by binding with coenzyme A. To complete this process, alpha-ketoglutarate must also be oxidized by NAD+, which is reduced to NADH. CO2 is removed as waste. In this step, succinyl-CoA loses its coenzyme A to become succinate. This reaction is preceded by several phosphorylations. A phosphate from the mitochondrial matrix displaces the coenzyme. Guanosine diphosphate, or GDP, seen here, is phosphorylated into GTP, or guanosine triphosphate. After this phosphorylation, the phosphate group is transferred to ADP, here at the bottom, to create ATP. Another ion from the mitochondrial matrix, called FAD, oxidizes succinate. This forms another energy coenzyme, like NADH. This energy coenzyme, however, is called FADH2. This also results in succinate becoming fumarate. In step 7 of the Krebs cycle, water is added to fumarate. This causes fumarate to become malate. In the last step of the Krebs cycle, malate undergoes one more redox reaction with NAD+. This creates NADH and oxaloacetate. After this step, the Krebs cycle is ready to begin again. ETC, or the electron transport chain, is the last step of cellular respiration. This occurs in the inner mitochondrial membrane and requires NADH and FADH2 created from the Krebs cycle. ETC makes up a majority of the ATP synthesized in cellular respiration and occurs in 3 stages. In stage 1, 2 NADH are created. In stage 2, 2 more NADH are created. And in stage 3, 6 NADH are created and 2 FADH2 are also created. There are 3 important terms associated with ETC. The first is chemiosmosis. Chemiosmosis is the movement of ions from an area of high concentration to an area of low concentration through a membrane protein. This results in the release of hydrogen ions, which are used to create ATP. The second term to remember is mobile electron carriers. These carry electrons between proteins in the inner mitochondrial membrane. The third and final term to remember is electron carriers. These are substances like ubiquinone and cytochrome C, which carry the electrons they received through the oxidation of glucose. There are 5 main components of the electron transport chain. The first one, or complex 1, is called NADH dehydrogenase. This is a transmembrane protein. Complex 2 is called succinate dehydrogenase. It is a peripheral membrane protein. The third component of the electron transport chain is called cytochrome BC1 complex. This is also a transmembrane protein like complex 1. The fourth component is called cytochrome oxidase, and the fifth component is oxygen, or the terminal electron acceptor, due to its high electronegativity. As you move down the electron transport chain from component to component, electronegativity increases. This means that the pull on electrons of the first component is very weak, while the fifth component can pull on the electrons with a very strong force. Energy in the form of electrons are dropped off at the first and second components of the electron transport chain. NADH drops off its 2 electrons at the first complex, whereas FADH2 drops off its electrons at the second complex. The movement of electrons through the membrane are taken care of by the 2 mobile electron carriers, UQ and cytochrome C. Ubiquinone, or UQ, carries electrons from NADH dehydrogenase to cytochrome BC1, complex 3. Cytochrome C then carries the electrons from the third component to the fourth component, cytochrome oxidase. FADH2 passes through fewer complexes of the ETC than NADH, therefore it creates 1 less ATP. As the electrons leave cytochrome oxidase, they combine with oxygen and hydrogen ions to form H2O. This creates the electrochemical gradient, which is required to power ATP synthesis. This is where chemiosmosis is introduced. Hydrogen ions in the intermembrane space found here are forced through the ATP synthase embedded in the inner membrane. This flow of hydrogen ions provides the energy required to create bonds between ADP and an inorganic phosphate through oxidative phosphorylation. This picture shows the theoretical yield of ATP from each step of the cellular respiration cycle. As you can see here, after pyruvate oxidation, only 12 ATP of the 36 has been created. It is in the Krebs cycle that most of the ATP is created. 2 of the products of the Krebs cycle are NADH and FADH2. In the Krebs cycle, 6 NADH and 2 FADH2 are created. In the electron transport chain, each FADH2 can yield 2 ATP, and NADH can yield 3 ATP. What this means is a whopping 24 ATP are created from the Krebs cycle alone, including the 2 ATP created inside the cycle itself. This adds up to anywhere from 36 to 38 ATP created from one cycle of cellular respiration, depending on how efficient the cycle was.